Pronounce a word, Sid. Polar bear, you say? Why? All right. TK to YouTube. All right, guys. Well, welcome back to my returning students. I'm happy to have you guys back in class for semester 2. And then to all my new coming students, welcome to my videos for the first time. So as I kind of mentioned in class earlier this week, I record videos, me just going through all the stuff that we talked about. So that way you guys have a way of reviewing everything, going over the learning objectives, so that way you guys feel a little bit more prepared going into our quizzes or our celebrations, as I like to call them, our celebrations of knowledge that we have on Mondays and Tuesdays for my classes. Okay, so the way this works is I try and break this down, not into all one video. As you see, I'm going to try and break down, especially this lab, into a couple different components. So what I'm going to start with is just going over the learning objectives, going through the pre-lab prep. I'm not going to go through, I'm not going to type out everything. I already have it all typed out just because that's how I presented it to you guys in class. And then, yeah, we'll talk about the stains, what blood staining looks like on slides, and then blood types as well. So we're going to try and break it apart into different pieces so that way it's not all combobulated into one giant, whatever, long video, okay? So 10 learning objectives this week. A lot of them are really easy, and you get 3 of them out of the way within the pre-lab prep, so it goes really, really quick. So the first 3 we'll do in the pre-lab prep. Number 4 is identifying each of the formed elements on the slides, images, and the blood cell models that we had, and then stating the relative abundance of each formed element, as well as classifying leukocytes. So I'm going to try and get through this within the first video, if possible, okay? So as you guys know, we have pre-lab prep every week. And this week I was lenient just because it was the first lab, so I just wanted to fill it out for you guys. So I also did that here for you too. So just zoom out here. So this is our first learning objective, okay? Describe the general properties of blood. So very, very straightforward, okay? Get my little marker to draw here. So if you can answer and if you can rattle off these characteristics and what the typical values would be for a healthy adult, you're going to be just fine with whatever question we throw at you asking about whatever is here, okay? So when it comes to blood, males and females have a total volume. Okay, males typically run a little bit higher just because they're typically a little bit bigger in size, a little bit taller, a little bit more muscle mass, have more fat, whatever. So they typically have a little bit higher blood volume. So typically about 5 to 6 liters. Females about 4 to 5 liters because of this. Okay, but pH values across both male and the female sexes are about 7.35 to 7.45. So it runs a little bit basic, but it tips itself right on that neutral line, where neutral is 7. So if you're swinging anything above 7, you're just a little bit basic. So just a little bit on the basic side, all right? The osmolarity value for both male and female sex, once again, is 300 milliosmolar per liter. It could be roughly around this number. They have a very specific number in my key here, but they give me an estimated number you can put down and just say roughly 300 to keep it nice and easy, all right? And then the hematocrit for males, typically, once again, runs a little bit higher, about 45 to 52%. And then for females, it's around 37 to 48%. All right, so number in this ballpark, if you can get it within either of these ranges that it gives us, you're golden if we throw a question at you asking about hematocrit, okay? So that's first learning objective right there, okay? Describing general properties of blood, and it says table 1.1, table 1.1 here, easy peasy, all right? So scrolling down here, this is second learning objective, okay? Knowing all the definitions for table 1.2 plus a sub-learning objective, number 3 here, talking about hemopoietic stem cells, all right? So first of all, these terms, what do we got? So the general term that we use for the production of blood, especially in formed elements, is hemopoiesis, all right? If you see a poiesis at the end of any of our words, it means the production of something, okay? We just got to know what the prefixes are to know what we're producing, all right? So hemopoiesis, hemo meaning blood, so it is the production of blood, all right? Especially the formed elements, that being red blood cells, white blood cells, and also platelets, all right? So the little sub-learning objective here, explaining the significance of the hemopoietic stem cells. HSCs is their other abbreviation, okay? So this learning objective is that all formed elements originate from HSCs, all right? So if you just know what the significance of HSCs are, you guys are going to be golden on that question too, all right? So all you got to know is all formed elements originate from HSCs, Very, very straightforward, all right? So 3 formed elements of blood here, okay? That being erythrocytes, leukocytes, and platelets. All right, so we have 3 things we got to talk about. Erythrocytes is our fancy term that we use for red blood cells, so RBCs, okay? Is our abbreviation, RBC. So we have erythropoiesis, all right? So once again, poiesis means the production of something, and since we have erythro in the name, it means the production of erythrocytes or red blood cells, all right? So if we're going to have an excess or a deficiency of these, we have a couple terms. So polycythemia, poly meaning many, all right? Cy meaning cell, themia meaning blood. So essentially having an excess of blood cells, red blood cells in particular, right? And then anemia is having our deficiency of red blood cells. So anemia is a common condition I'm sure many of you have maybe heard of, or maybe you've had it. Maybe you've had it in the past life or a past chapter of your life. So it's very common to see in a lot of female cross-country runners. If they're having alterations of their diets, if they're on their menstrual cycle, really, really common to have anemia in that patient population as well, all right? And then we have sickle cell disease down here, okay? So this one's really, really interesting. So this is a hereditary condition in which a mutation in hemoglobin causes red blood cells to sickle when oxygen is low, okay? So when oxygen is low, so typically you could see this when you're up at a higher mountain range, so at a higher elevation or higher altitude, okay? So with these patients, okay, when you get to those higher elevations, your oxygen levels are depleted. So what's going to happen is the red blood cells will sickle, so they turn into that weird sickle cell shape. And I kind of had that picture in class, but it kind of has like a handle down here. It belongs to like the USSR flag, and it has that kind of crescent appearance to it. So they kind of take on that shape. Now that's a perfect world, as you guys saw on the slides. Some of them have like 4-leaf clover looking appearances. Some of them look like ovals. Some of them look like rectangles. But if this is a perfect world, what a sickle cell should look like for a red blood cell, this is what it would look like roughly, okay? So this mutation is predominantly found in African Americans. And the reason being is because malaria is a huge illness and disease that is found in Africa. So this hereditary mutation actually allowed for those that have this sickle cell traits or disease to be able to be immune to malaria. So it prevents it from taking place. And how? I don't really know the immunology behind it. That's really not my scope of practice. You could ask my fiance. I'm sure she give you a whole rundown on that. She just studied her butt off for a big exam talking about that. Okay, so that's red blood cells. Then we have leukocytes. Okay, right after this. So leuko means white in Greek. Okay, and then cyte meaning cells. So it is simply just white blood cells. Okay, leukocytes, white blood cells, WBCs. All right. And then, so leukopoiesis. Once again, poiesis, the production, leuko, white, the production of leukocytes or white blood cells. Very, very straightforward. Leukopenia means to have a low white blood cell count. And then leukocytosis is having a high white blood cell count. All right. So if you have a high white blood cell count, more than likely you either have some sort of an infection, a bacterial infection, you have some sort of an illness like a virus or something, cold or flu. If you ever went to go and get your blood drawn when you have either of those conditions, for example, normally you'd have a higher white blood cell count because you're trying to fight off illness, infection, etc. All right. Excuse me. And then our last one here is leukemia. So this is a cancer. I'm sure some of us or many of us have heard of it. It is a cancer of the hemopoietic tissues that produces excessive amounts of immature leukocytes. All right. So what does this mean? So if we're producing in excessive amounts of these leukocytes, but they're immature, one, they're not going to know what to do. Okay, so you're producing a whole bunch of these things that are basically children. You're asking these children to do something very, very complicated to fight off illness, fight off whatever. Well, one, they're not going to know what to do. They might do the right thing, they might do the wrong thing. Okay, so this leukemia, this cancer condition, is very deadly because your white blood cells don't know what the heck they're supposed to do, so they can't really fight off infection, disease very well. And eventually it'll end up killing you, okay, if you don't get it treated. So it's a very, very deadly form of cancer that you can get, all right? And then we have platelets down here, okay? So platelets, it's a it's the second most abundant form element. We'll talk about the abundance, but what we use platelets for is we use it for clotting, so for helping to stop bleeding, essentially, with our blood. So we get a paper cut, we get a cut, we cut our finger, whatever, it's going to bleed. Our platelets are what is primarily responsible for stopping that bleeding so we can clot our blood so we don't bleed out, All right, very, very important thing to have in our blood. Thrombopoiesis is the term that we use for the production of platelets. All right. Hemostasis is the actual process of stopping bleeding. It's the actual point at which bleeding stops. Is called hemostasis. We have these other two fancy terms down here called a thrombosis and an embolus. All right. Thrombosis is the abnormal clotting of blood in an unbroken vessel. All right. So it's where a blood clot occurs where it should not be occurring, essentially. All right. It's a very common thing to see in the lower leg, particularly in the calf. We typically call that in medicine a deep vein thrombosis, okay? Something that is a little more common after someone that gets a knee replacement. We typically have those patients go on blood thinners for a while to try and prevent this from happening. But it is a possibility. And if it does happen, and for some reason, if that clot, that thrombosis breaks loose and starts traveling throughout the bloodstream, you're going to get something called an embolus, and then if that gets up into the lungs, you can get a pulmonary embolism as a condition. All right, so that is another urgent, potentially life-threatening condition that must be addressed urgently. All right, so learning objective, table 1.2, sub-learning objective, number 3 here, talking about hematopoietic stem cells. All right, so that's the pre-lab prep. That is 1, 2, and 3. Perfect. So let's scroll down here. All right, so we chatted about the components of blood. All right. So if you wanted to identify all of those together, okay, if you wanted to separate them out more specifically, you could use a centrifuge to spin them down. So I work a lot with a physician at my practice who does PRP injections, so platelet-rich protein injections. You take out someone's blood, you spin it down, it looks like this. What you're trying to get at when you're doing these injections is this buffy coat, because that's where the platelets lie, all right? You get that, and it has a lot of healing factors on the inside of it. So you're trying to inject that buffy coat back into someone's, whatever, patellar tendon, quad tendon, plantar fascia to help aid with healing. All right, it's a kind of experimental treatment. But if you wanted to actually separate the blood into its separate components, you could use a centrifuge. And there's a lot of medicine components throughout this that can be used in orthopedics, especially. All right. So I remember you guys had to fill out this table. I'm gonna fill this one out with you guys together here, okay? Walk through it together. So the average adult male, remember we talked about this. All these answers are found in the tables 1.1 and also 1.2. So the average adult male, remember, has about 4 to 5 liters of blood, or 5 to 6, I believe, if I remember correctly. 5 to 6, excuse me. But if you say 5, that's fine too, okay? 5 liters is plenty to say here. So blood plasma, the liquid portion of blood, comprises about 55% of the blood volume. In a healthy adult, plasma is composed of water, electrolytes, ions, gases, protein, etc., etc., etc. Okay. The pH of blood plasma must be kept within its narrow limits. Remember, this is going to be 7.35 to 7.45. Let me just add a space here. 7.45. Perfect. So that the proteins and enzymes in the blood and surrounding tissues remain functional. Because if we were to alter this value, so if we get more basic or get more acidic, that's when we can start to see the breakdown of those proteins and enzymes. All right, we don't want that to happen. The osmolarity of blood is constantly monitored by the brain, and if necessary, salt or water are excreted by the kidneys. We'll talk about this more when we get to the urinary system, which will be later on, way after spring break, so way in the future. So we try and keep this value roughly around 300 milliosmolar per liter, right? All formed elements of blood originate from hematopoietic stem cells. All right, remember this is one of our learning objectives. Through a process called what? Remember, guys, this is going to be called hemopoiesis, all right? Hemopoiesis, all right? Perfect. Erythrocytes, or red blood cells, are the most abundant formed element. Okay, we're starting to talk about abundance. This is one of our learning objectives, all right? So take note of this. This is the one takes the crown, most abundant. The percentage of whole blood volume composed of erythrocytes is called the hematocrit. Okay, so hematocrit, and this usually roughly runs around 45%. You can also add 52% in there too. Remember, just be in the ballpark, about 45 to 52% in men. Okay. The primary function of erythrocytes is to transport oxygen and carbon dioxide. All right, this is made possible by a special molecule called hemoglobin, which contains ions that bind oxygen because of a high affinity that they have towards it. All right, erythrocytes are formed from HSCs by a process called what, guys? Remember, it's a poiesis. What's the Greek word for red? Erythro. Erythropoiesis. All right. If red blood cell count is too high, a person is said to have what? This is called polycythemia. All right. In contrast, a significant deficiency, in red blood cells is called anemia. All right. Anemia is that. And then blank is a hereditary disease that occurs mostly in people of African descent, as I said, okay? So, what is this one called? This is called sickle cell disease, all right? It is caused by a recessive allele that encodes for a form of hemoglobin that does not bind to oxygen very well. So when it is deoxygenated, aka you're at a higher altitude, so up in the mountains, for example, this hemoglobin causes red blood cells to become deformed into a sickle shape, which then blocks blood vessels and can cause intense pain. All right. So if this were to continue, for anyone who's interested, my athletic training student who I know is in this class, you can get a condition called rhabdomyolysis. All right. It'll be something that might show up on your board of certification exam in the future. So next one. So remember, platelets, second most abundant formed element, although they are far less numerous than erythrocytes. Okay. And remember, we saw that on the slides, how abundant the blood, they're everywhere, the red blood cells. The platelets look like those little polka dots like we were talking about. Platelets, though, all right, what these guys are formed by the HSCs by a process called thrombopoiesis, remember, okay? Poiesis. And they play a major role in blank by causing vasoconstriction. So what is this going to be? This is going to be hemostasis. Hemostasis, beautiful. So most strokes, heart attacks are due to blank or the abnormal clotting of blood in an unbroken vessel. So this one is going to be a thrombosis, and a blank. And if a blood clot breaks loose and starts traveling through the bloodstream, it is called a embolus. All right, lost my word there. My line I was reading, embolus. So this could be extremely dangerous since the clots may lodge in a small vessel supplying blood to the heart, lung, or brain. All right, that's how heart attacks happen, as it was saying here, or strokes. All right, leukocytes, or white blood cells. They are the least abundant. All right, remember, they're broken down into a bunch of different types of leukocytes, which we'll talk about in a second. They are the least abundant formed elements, but there are many different types of leukocytes that play unique roles in protecting the body against infection and disease. All leukocytes are formed from HSCs by a process called, once again, leukopoiesis. All right. In certain diseases, measles, mumps, polio, AIDS, for example, all right, the white blood cell count can reach dangerously low levels, leaving the patient at serious risk for infection. All right, and this condition is called leukopenia. All right, an elevated white blood cell count is going to be called what? All right, so this one is going to be called leukocytosis. This may indicate infection, allergy, or some other condition in your blood if this is elevated, all right? It is useful to have differential white blood cell count performed to determine which type of leukocyte is elevated. Because each type of leukocyte plays a specific role in immunity, knowing which cell levels are elevated might provide clues, rather, as to if a condition is caused by bacteria, virus, parasite, or an allergen, for example. Cancer of hemopoietic tissues can produce a condition called leukemia. All right, that cancer condition, where the number of leukocytes and their precursors is extremely high. This can be especially dangerous because these immature cells replace normal bone marrow, and person suffers from a deficiency of normal leukocytes, erythrocytes, and platelets. Patients are therefore vulnerable to opportunistic infections, anemia, and hemorrhage at the same time. Okay? All right. So you filled out all this stuff. All right. Now we're going to break down these leukocytes. All right. So how are we going to break these down? So remember, leukocytes, guys, they are broken down. Let's see here, leukocytes. All right, they are broken down into two components. All right, let's see here. They are broken down into granulocytes. Okay, and then they have agranulocytes. All right, let's talk about the granulocytes quick. So, granulocytes, what are they broken down into? So if they're granulocytes, it means they have specific granules that absorb stain and become conspicuously visible under a microscope, okay? These include neutrophils, eosinophils, and basophils, okay? I'm going to put these in order from that mnemonic I taught you guys, okay? So I'm going to start with B, if you guys remember it. So the first one is going to be basophils. All right. So basophils is the least common, all right, as it indicates here. Our next one that comes right after this, remember it starts with an E, is eosinophils. And then what's our one that comes right after this one? Starts with an N. This is the neutrophils. Okay. And remember, what is our mnemonic that we use for this guy? Okay. Remember, if it has granulocyte, okay, it's gonna be a G. You have the G here, okay. But then BEN in that order, we have Ben-G, right? Really bad hand. I'm writing on the mouse here, so apologize for this, guys. Ben-G, where the G represents the granulocytes, okay? So B, basophil, E, eosinophil, and neutrophil. Furthermore, guys, remember, all the granulocytes, I told you this, they all end in this suffix. They all end in phils, all right? If you see phils in the suffix, that means it is a granulocyte, okay? So if you're wondering which one is which, if it ends in phils, it's a granulocyte, okay? So it has the granules. What's the next one? So we have the granulocytes, but then we also probably have the agranulocytes. So A in Greek means without, okay? So that means that these do not have granules on them, okay? Let's get this guy lined up, perfect. So we have granulocytes here. So I'm going to do this one, same thing, same order that I did, all right? So it's going to start with an M, monocyte. And what's the one that's right below that? Starts with an L. This is lymphocyte. All right, perfect. So remember, what's our mnemonic for this one then, okay? Remember, MAL, Spanish word for bad, but we use the A here, or our A granulocyte. And then we use the M and the L, so we have monocyte and lymphocyte, okay? Furthermore, remember, granulocytes end in phils. Agranulocytes end in cyte. C-Y-T-E, okay? So if you see cyte in the end of the name, okay, exception of agranulocyte and granulocyte, that means it is going to be a part of this category for the agranulocytes, okay? So, MAL, M-A-L, all right, that's your mnemonic here. Ben-G, that's your mnemonic for the granulocytes, okay? All right. Not going to talk about functions on this page. I'm going to talk about abundance real quick because it's all labeled right here, and I want to get you guys that mnemonic we talked about, okay? So remember, what's the order? All right? Remember, from top to bottom, starting with most up here, okay? So the most abundant right here. And then we're going to have least all the way down here, okay? Perfect. So what's our mnemonic that we have? Remember, the first one always takes the crown is erythrocyte. All right. Directly below that one, we're going to have platelets, all right, as labeled up above. And now we get to leukocytes. Remember, this is where things get a little confusing because there's like 5 different leukocytes we have to talk about, right? And they all have different abundances in the blood. Here's how we do it. All right, remember the mnemonic I taught you? All right, it is never let monsters eat biology. And if we match up those first letters, they match up to all of our leukocytes. And it goes in this order, okay? That direction. So, never. What does that match up with? That is neutrophil. All right? So that is neutrophils right there. Number 4, never let. All right, that is lymphocyte. Okay? Number 5, monsters. That is going to be our monocyte. Number 6 is going to be our eosinophil. And then our last one here, last but not least, is going to be our basophil in this order. Okay. All right. So never let monsters eat biology. And this is just leukocytes, okay? Remember, erythrocyte, platelets, that's easy, all right? Erythrocytes always first, platelets always second, okay? After that, never let monsters eat biology in that order. If you know the name, all right, you know what order they're going to be in by abundance, okay? So if we throw you a question next week, like we give you 3 or 4 of these, and we ask you to label their abundance in order from most to least, we should be able to do that with this mnemonic and knowing that it's always erythrocyte first, platelet second, every single time, okay? Okay, so I'm going to stop here. Next video, I want to start working on this table with you guys. All right, we're going to fill this guy out, talk about classification, appearance, formation process, abundance, function, all that stuff, okay? So, before we sign off here, I want to recall to you guys, remember, this is a learning objective. Knowing the breakdown of which ones are granulocytes, which ones are agranulocytes, okay? Another learning objective right here, the relative abundance of these, okay, from most to least or vice versa, all right? So I scroll all the way up to the top here. I'm going to highlight what we've done. So we for sure did 1.1 together. We talked about the key terms and conditions, table 1.2. Talked about the significance of hematopoietic stem cells, HSCs. All right, we'll talk about number 4 in the next video. We stated the relative abundance. All right, we're going to name their functions in the next video, so we'll keep that one unhighlighted. And then we classified leukocytes as granulocytes or agranulocytes. All right. So next video, we're going to tackle 4. We're going to finish number 5. We're going to do number 7, and then we're going to move on to blood types. All right. So I'll be back in a second.